Enzyme catalysis. Let's look at the contents of this presentation. First, we will define what is catalysis. Then, we will look into what is enzyme catalysis, characteristics of enzyme catalysis, principles, and mechanism of enzyme catalysis. So, catalysis is the process through which the rate of a reaction is altered while the quantity and chemical properties of the catalyst remain the same. It can be defined as the increase in the rate of the chemical reaction due to the presence and action of an additional substance that is called as a catalyst. The catalyst is not consumed in the catalyzed reaction and can continue to react repeatedly. Usually, even a small amount of the catalyst is sufficient to bring about this effect. The catalyst reacts to form a temporary intermediate which then regenerates the original catalyst in the cyclic process. So now, what is the action of enzymes? Or what is the role of enzymes in catalysis? Enzymes refer to a category of catalysts assisting in accelerating and stimulating a number of crucial metabolic events in animals and plants. The process of enzymes binding to a substrate and then increasing its conversion rate to products is called as enzyme catalysis. The site of the enzyme that interacts with the substrate is called the active site. The enzyme interacts with the cofactors or coenzymes to bring about catalysis. And further, an enzyme enhances the rate of both the forward and backward reactions, thus maintaining the chemical equilibrium of the system. Now, looking at the characteristics of enzyme catalysis. A single molecule of enzyme catalyst can transform up to 1 billion molecules of the reactant per second. Hence, enzyme catalysts are said to be highly efficient. These biochemical catalysts are unique to certain types of reactions. That is, the same catalyst cannot be used in more than one reaction. The effectiveness of a catalyst is maximum at its optimum temperature. The activity of the biochemical catalyst declines at either side of the optimum temperature. Biochemical catalysis is dependent also upon the pH of the solution. A catalyst works best at an optimum pH which ranges between 5 to 7 pH values. Also, the activity of the enzymes usually increases in the presence of a coenzyme or an activator such as sodium plus, that is Na plus, or CO2 plus. So now, looking at further at enzyme catalysis, it can be considered as a process by which there is an increase in the rate of a reaction through a biological molecule called as an enzyme. Here, the enzymes can increase the speed of the reaction but cannot affect the equilibrium position. We also have to note that the site of the enzyme that reacts with the substrate is called as the active site. Also, we see that enzymes can only reduce the energy barriers between products and reactants, and hence one of the most important principles is that enzymes always catalyze reactions in both the directions. So, looking at the principle of enzyme catalysis, it is that enzymes increase the rate of a chemical reaction without being consumed or altering the chemical equilibrium of the reaction. Catalysis by enzymes in a cell is vital because most biochemical reactions have meager rates of reactions when uncatalyzed. So an uncatalyzed, or where a reaction does not have a catalysis, then the biochemical reaction, those biochemical reactions will have a smaller or meager rate of reactions. The vital factor is the reduction of energy, which acts as a barrier between the reactant and the products. Reducing what is called as the activation energy increases the number of reactant molecules that can overcome the barrier and hence get transformed into a product. Therefore, in a chemical reaction, the reactant molecules should contain optimum energy to cross the energy barrier, that is activation energy. Though the reactant molecules do retain varying amounts of energy, only a few of them have enough energy higher than activation energy. Enzymes function by decreasing the activation energy through the formation of what is called as an enzyme substrate complex. Also, it is to be noted that there is a small cavity on the surface of the enzyme which functions as the enzyme or the active site. This is the place where one or two substrates, that is the reactant molecules A and B, or A alone or B alone or A plus B, can bind. The flexible enzyme changes its shape when the substrate binds to it. This is one kind of a hypothesis. The substrate and the enzyme form a substrate-enzyme complex. The changing in shape can be because of different kinds of models which has been researched. Which also means that we now look into the mechanisms of enzyme catalysis. The first one is called as a lock and key model. This is one of the kinds of hypothesis showing how enzymes and the active site are brought together during a enzyme catalyst reaction. Enzymes have defined active sites, as mentioned earlier, that are capable of substrate specificity. They are specific. One of the hypothesized mechanisms of enzyme catalysis is called lock and key model. Here, the reactant molecule or the substrate fits into the crevice like lock and key. The next one is called as an induced fit model. This theorizes that simple reaction between enzyme and the substrate is fairly weak. So, the initial reactions can rapidly cause conformational changes in the enzyme that make the binding between the enzyme and the substrate stronger. The substrate comes closer to the enzyme surface and induces a change in its shape, leading to the correct alignment of the substrate and the enzyme. The catalytic groups at the active site react with the reactant molecules to form the products which separate from the surface of the enzyme, and then the process is repeated. In addition to these two models, we also have the alternate routes of enzyme catalysis, such as proximity and orientation of the enzyme substrate complex, proton donors or acceptors to neutralize the charges during the transition phase or transition state, electrostatic catalysis for stabilization of the activated complex, covalent analysis for lowering the energy of the transition state, and metal ion catalysis in the active site.